Hello everyone, myself Shonok again with you all. That at the previous lecture we have discussed about that what is a respiration is. So everybody know that what is a respiration is nothing but the process through which a living organisms used to respire or is a formation of the counting of the energy, that is the ATP. It's a process that deals with the formation of the ATP. So we all know that there are two kinds of organism which are able to form the energy of their own, that is known as the photosynthetic organism. We have discussed earlier photosynthetic organism, and another one who are not able to basically form the ATP of their own, that is the non-photosynthetic organisms. We have learned about the at the previous lecture. These are the two kinds of organisms, and we know that respirations are of two types. One is the oxygen dependent, another one is the oxygen independent, that is the O2 dependent and O2 independent. In case of O2 dependent, it is known as the aerobic respiration, and in case of O2 independent, it is known as the anaerobic respirations. So we have dealt with these are basic things, that what are the particular energy source that we are using. So if we recall at the previous class, what we have did is the energy source or the substrate or the particular molecule that we are taking to extract the ATP out from it is the nothing but the glucose molecule in account. That is C6H12O6 plus, in if I consider as a aerobic respiration, in presence of O2 yields us 6CO2 plus 6H2O plus ATP, and this ATP is being utilized by the most of the organisms. So we dealt with what we have learned about at the previous lecture. And so what is the 6CO2 and 6H2 over here is nothing but the final product. And in case of aerobic respiration, who is the final electron acceptor of the total reaction? That is nothing but the oxygen molecule. So the oxygen molecule is the final electron acceptor of the aerobic respiration. So we have learned about this, what is the aerobic respiration, what is it. And so let's move directly into the one of the famous process of the aerobic respiration. You may say it occurs in both aerobic and anaerobic, but we will consider it to be aerobic respiration because in our syllabus only aerobic respiration is there, that is the glycolysis. So first deal with what is the glycolysis. So see the name, if you see the name carefully, the glycolysis means, this glyco means glucose, and lysis means to break down. We all know, to break down. So what actually happens in this process is the breakdown of glucose occurs to gain is nothing but the ATP, and this ATP is the energy currency of the cell. We all know. So glycolysis is the process of breakdown of glucose to generate ATP. So this is a catabolic process. Now, what is a catabolic and anabolic? Let's discuss a very few points. That if we know, if we want to break down any kind of molecule of any kind of substrate, for example, to gain ATP, we are breaking down any kind of substrate or molecule to gain ATP. If we break down any kind of molecule, that is known as a catabolic reaction. Catabolic reaction. And in contrast, if we basically again rearrange or altogether all of the small pieces of molecules to combine and form a huge molecule, that is a anabolic reaction. So in case of glycolysis is a catabolic reaction because we are breaking down glucose molecule to obtain the molecule of our interest, that is the ATP, that is the adenosine triphosphate, and is the most energy source for every living cells, we all know. So this is our basic denotation that I can say that what is a glycolysis is. And again, it is also known as the Emden Meyerhoff pathway. And number two, it is a catabolic process. Sorry, it occurs in both aerobic and anaerobic conditions. So, what is the output of this glycolysis? That is ATP. How many molecules that we basically that is the two molecules of ATP. So this is a overview of a glycolysis that I can basically see that this glycolysis is also known as the Mden-Marov pathway, and most of the competitive examinations is this pathway is being asked instead of glycolysis. So I have also mentioned the important points, and it's a catabolic process. Both are aerobic and anaerobic conditions, and output is the two molecules of ATP as a energy source, and another one there's two molecules of pyruvate. So how this ATP or how this pyruvate is occurring? So without wasting any time, let us move directly into the pathway that how a glycolysis is being occur. So let's move. So understanding the pathway. So our main substrate of this glycolysis is glucose. This glucose is being break down to first step, that is the glucose 6 phosphate. Okay, so glucose is being converted to glucose 6 phosphate under the action of the enzyme hexokinase. So first step is the glucose to glucose 6 phosphate under the action of this hexokinase, and we all know in the enzyme kinetics that many of the enzymes work best at with the metal enzyme, with the metals, and some of them are Mg2 plus, Mn2 plus. There are lots of metals, and the enzymes are known as the metal enzymes because it works under the influence, or so most of the enzymes basically works in with the collaboration with the metals, that is one of the metal enzymes. And the first enzyme that helps in the conversion of glucose to glucose 6 phosphate is the hexokinase. And whenever there is a possibility of hexokinase, this kinase refers to ATP. So at the first step, we give externally the ATP, where this ATP is converted into ADP under the action of hexokinase. Wherever there is a kinase molecule, just first remember that there is a action of ATP in the conversion to ADP. So next, this glucose will be converted to glucose 6 phosphate. Next, this glucose 6 phosphate will convert to fructose 6 phosphate under the action of, and this glucose and fructose are isomer of each other. Then what will be the name of the enzyme? That is the phosphofructokinase. So first is the glucose to glucose 6 phosphate, next the glucose 6 phosphate to fructose 6 phosphate. Next, this fructose 6 phosphate will convert into fructose 1,6 bisphosphate. Okay, and the enzymes that helps in the regulation from fructose 6 phosphate to fructose 1,6 bisphosphate is the phosphofructokinase. And again, this fructose 1,6 phosphate will convert into glyceraldehyde 3 phosphate. So I have break down this glycolysis into two parts. So see, first is the glucose as a substrate, next glucose 6 phosphate, next fructose 6 phosphate, next fructose 1,6 bisphosphate, and 5 is the glyceraldehyde 3 phosphate. So there are total 10 steps in glycolysis, and out of which this 5 steps is known as the investment phase or preparatory phase. Preparatory phase. Why this preparatory phase? Because we all know that in case of preparatory phase, we are basically investing ATP to gain out at a maximum amount. So in any kind of business, we all know that first of all investment is necessary. So similarly, in body, whenever there's a glycolysis occurring, at the first 5 steps we invest ATP. Then the question may be asked that how many ATP, how many molecules? See, first of all, when there is a possible conversion of glucose to glucose 6 phosphate, there is an action of hexokinase. And I have said that whenever there is a kinase, there will be a ATP molecule will be invested. So similarly, only one molecule of ATP. Again, so you can see that from glucose 6 phosphate to fructose 6 phosphate, that is the enzyme is the phosphofructo that is the phosphofructo isomerase. So they are isomers of each other, so there is no kind of ATP molecule. And next you can see this fructose 6 phosphate to fructose 1 6 bisphosphate. As you can see, the enzyme is the phosphofructokinase, and this kinase is nothing but the action where ATP molecule is converted to ADP, and where there is a investment of ATP. So number one molecule, number two molecule. So in the preparatory phase or the investment phase, we basically invest two molecules of ATP. Two molecules of ATP. So first is the glucose to glucose 6 phosphate. The enzyme is the hexokinase under the action of Mg2 plus and the Mn2 plus. Next, glucose 6 phosphate is converted into fructose 6 phosphate under the action of the phosphofructo isomerase because they are isomer of each other. Next, phosphofructose kinase will convert into fructose 1,6 bisphosphate because they under the action of the phosphofructokinase, and this kinase represents ATP, we all know. So there are two ATP molecules are invested at the preparatory phase up till the and at the last the glycerol 3 phosphate occurs. So this is the first initial pathway I have discussed. So let's please you can resume the screen and pause the screen, and again basically you take a snapshot or you may write in your notebook. So moving without doing further delay, let's start the rest of the step. So we have discussed till far that this glucose is being basically converted into glucose 6 phosphate, then fructose 6 phosphate, fructose 1 6 bisphosphate, then glycerol 3 phosphate as a last entity. Then what will happen to this glycerol 3 phosphate? So let's take a look. Glycerol 3 phosphate. This will convert to DHAP. We all know that is the dihydroxyacetone phosphate. And this glycerol 3 phosphate and this DHAP are isomer of each other, and under the action of the enzyme triose phosphate isomerase. Enzyme is the triose phosphate isomerase. So glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 bisphosphate, glycerol 3 phosphate, and they will just basically convert into the DHAP under the action of triose phosphate isomerase. And we know basically whenever we are investing some amount, for example, we are investing 5 rupees in a particular business. So we always want this 5 rupees to get double or more than that. It may be 10, it may be 20, it may be nothing, it may be anything. So in case of glycolysis, we are obtaining ATP. So we have invested how much ATP? That is a 2 ATP. So if we take a return of 2 ATP only, then anything profitable comes into account? The answer is no. So basically, our body is so much schematized and so much engineered so highly that this glycolysis occurs in two multiplicative phase. For example, One is coming from glucose, 2 ATP is being done, and this glyceraldehyde 3 phosphate will basically carry out the rest of the step. Again, this DHAP will carry out the same step of the glycolysis. So whatever the end product we are getting, it will be multiplied by 2. So similarly, if we invest 2 ATP, then the net ATP will be 2, because for 1 ATP 1 ATP is coming, for 1 ATP 1 ATP is coming. But if we get this 2 ATP as a return, then there will be no kind of energy gain. So as our body is so much highly biologically modified, it always run glycolysis into two parallel pathways, one from glyceraldehyde 3 phosphate, another one from the DHAP. So ultimately 2 ATP into 2 ATP gives us 4 ATP. So at the previous slide, I have discussed you that you may see that how much ATP molecule has been gained. You can see over here there is two molecules, and these two molecules are for each of the glycolytic cycle. That's for 2 ATP, 2 ATP, two molecules of ATP is generated. But our glycolysis occurs in two parallel pathway at the same reaction. So ultimately, it will be 4 ATP as an overall reaction. I guess you are uh, hope clear with this particular concepts. So let's move on to the next one. So so this DHAP will convert this DHAP will convert to 1,3 bisphosphoglycerate bisphosphoglycerate, and the enzyme will basically the enzyme will be nothing but glyceraldehyde 3 phosphodehydrogenase, because there's a dehydrogenation of this DHAP. And before the dehydrogenation, who is oxidizing this DHAP? That is the NAD plus. And this, as before conversion to this 1,3 bpgh, it will oxidize by the NAD plus, and after the conversion of 1,3 bpg, this NAD plus will convert to NADH. NAD plus will convert to NADH. Understood? So from THAP to 1,3 bpg. I hope it is clear. Then from this 1,3 bpg, it will convert to, that is the 3 phosphoglycerate. 3 phosphoglycerate. And this is the most important step from the point of view of question, because here the investment phase is over up till the fifth step, which I am discussed earlier. But now we are gaining of the ATP. So at this step, from the conversion of 1,3 bpg to 3 bpg, we are gaining out one ATP molecule. And how we are getting out this one ATP molecule? That is the direct phosphate group transfer from ADP to ATP. And when there is a direct transfer of ADP to ATP from high energy compound to yield ATP, this is known as the substrate level phosphorylation. Level phosphorylation. That what is the main step? That is 1,3 bpg to 3 phosphoglycerate. We get the substrate level phosphorylation occurs here. So this is the first substrate level phosphorylation. We are basically getting ATP molecule, and is the first step of glycolysis. Then from 3 phosphoglycerate we get the 2 phosphoglycerate, then phosphoenol pyruvate, then pyruvate, and this phosphoenol pyruvate to pyruvate is again we are gaining an ATP. This is the second substrate level phosphorylation. And all of these steps are irreversible. This DHAP to 1,3 bpg to 3 pg, this is everything but except the last step. And again at the last step there is a enzyme known as the pyruvate kinase. And where is the kinase? I have already mentioned earlier that wherever there is a kinase, there is nothing but ATP molecule. So This parallel pathway is will also run in this particular glycerol 3 phosphate also. So ultimately we are gaining 2 ATP. You can see this one and this one. So for one pathway you are getting 2 ATP. So for two pathway parallel pathway you will get 2 into 2, that is the 4 ATP. So from one molecule of glucose molecule in a glycolysis pathway we are gaining of 4 ATP. But if we omit one pathway, then for two molecules of ATP. So this is the main thing. So moving on to the next, the important points that I want to discuss with you for the exam point of view, that is the first is the enzymes hexokinase. You may note down this, use Mg2 plus and Mn2 plus for the action of their performance. Next, the first substrate level phosphorylation occurs in from glycerol 3 phosphoglycerate to, sorry, 1 3 phosphoglycerate to 3 phosphoglycerate. And the second substrate level phosphorylation occurs in phosphoenol pyruvate to pyruvate. And glycolysis occurs in cytosol. Okay. I have also already discussed this, and again point was is the, yeah, the glycerol 3 phosphate, sorry, the glycerol 3 phosphate is basically oxidized by, glycerol 3 phosphate is basically oxidized by NAD plus. Okay, so this is the important point. And how many molecules of ATP you are gaining? There is the 4 ATP molecules for one glucose molecule, because two parallel pathways are going on. And the last one is the 2 molecules of pyruvate we are gaining. So this is the main thing. I hope the idea is clear with you, and also the pathway. If you are have any queries, you may feel free to ask in the comment box, and I will hopefully try to attend all of your questions. And if some any other queries regarding the pathway, if you can also mention in the comment box, I will explain it again or make a different video for upcoming lectures, into the upcoming lectures. So these are some PYQs. Let us solve with some of the main problem. So the first question, C, which of the following statement is not true? Not true. We have to understand about not true. So it occurs in the cytosol of the cell. Okay. It also helps in fructose metabolism. Glycolysis generates ATP. Glycolysis generates CO2. So at the first go, we know that glycolysis generates ATP. Absolutely correct. Glycolysis generates CO2 molecule. Yeah, it's also absolutely correct. It occurs in the cytosol of the cell, so absolutely correct. So it is a not a fructose metabolism. So option B will be the correct answer. It is a basically a carbohydrate or a glucose metabolism. So option B is the correct answer. So let's move on to the next. So match the following with the enzymes with its substrate. Okay, fine. Hexokinase 1 will be the fructose 1,6 bisphosphate. Hexokinase basically converts glucose to glucose 6 phosphate, not fructose 1,6 bisphosphate. So number one option is absolutely wrong. So pyruvate kinase to 2 phosphoglycerate. So basically pyruvate kinase convert phosphoenol pyruvate to pyruvate. So option 2 is also wrong. So enolase convert phosphoenol pyruvate. Enolase convert phosphoenol pyruvate. Yeah. And number 4 is the aldolase convert glucose. No, glucose is converted by hexokinase. So these three options is absolutely wrong. Then the option C is the correct answer. If you don't know about the enolase also, basically if I have missed, this enolase helps in the formation of 2-phosphoglycerate, 2-phosphoenol pyruvate, then to pyruvic acid. 2-phosphoglycerate, to phosphoenol pyruvate, to phosphoenol pyruvate, then pyruvate. So this 2-phosphoglycerate to 2-phosphoenol pyruvate, this is done by the enolase, the enzyme. So if you don't know also, but you can try out the option elimination method. That is this one is incorrect, this one is, and this one. So option C is the correct answer. So hopefully my lecture is well comprehended to you all. So let's see you in the next lecture with other kinds of TCA cycle and other biochemical pathways we will discuss on the later slides and the later lectures. So till then, please take care of yourself. Have a good day.